Hello and Assalamualaikum. So for this video, our group gonna present about metagenomic in extremophile research. And here are the group members. And the research paper that we use for this presentation is metagenomic analysis of hot spring in central India, reveals hydrocarbon degrading thermophile and pathway essential for survival in extreme condition. And where we found this research paper is in journal named Extreme Microbiology.
These are the list of the authors of the paper, and the paper was published on January 5th, 2017. The authors are affiliated with two departments of Indian Institute of Science, Education and Research. Those departments are Department of Biological Science and Department of Earth and Environmental Science.
There are two techniques that used in this research paper, which is 16S rRNA sequencing and shotgun metagenomic sequencing.
Assalamualaikum, my name is Muhammad Haziq. Today I will explain regarding the 16S rRNA gene sequencing technique. So what is 16S rRNA sequencing technique? It refers to the sequencing of the 16S rRNA gene that codes for the small subunit of the ribosome found in prokaryotes such as bacteria and archaea. For better understanding, you can refer to the image on top right of the slide so that you can better understand where the 16S rRNA gene is located in the prokaryotic ribosome. Next, what is the function of 16S rRNA sequencing technique? This technique is typically used for identification, classification, and quantitation of microbe within a complex biological mixture, such as in environmental sample, such as sample from hot spring, which microbe is difficult to be cultured. So how does the 16S rRNA sequencing work? Firstly, we need to know that 16S contain both highly conserved region as well as hypervariable region. The conserved region, as you can see in the slide, indicate phylogenetic relationship among species and are used as sites for PCR priming, while hypervariable region indicates the difference between species. The presence of highly conserved region allow researchers to design the primer pairs that will accurately and reliably amplify the 16S hypervariable region of their choice.
Next, we are going to see the workflow for the 16S rRNA sequencing. This diagram shows the exact procedure for conducting this method. First, there are simple preparations. Second, library constructions. Third, sequencing. And finally, data analysis. For sample preparations, from culture bacteria or bacterial colony, we extract the DNA using the metagenomic DNA solution kit. We must make sure that the obtained DNA is enough for the next step. For the second step, after DNA solutions, PCR applications of the V3 and V4 regions are done using a bacterial primer pair. The main reason that we are using V3 and V4 region is because these regions contain the maximum nucleotide heterogeneity and it contains the highest differentiating strength compared to other regions. This will help us in classifying bacteria in the later analysis step. Then in the third step, the PCR product that we got from V4 are then used in the sequencing platform such as Illumina MySet method or Sanger sequencing method. Sequence products are then verified, quantified, and then pooled. This is to determine the sequence of nucleotide and the target bacteria DNA. And lastly, data analysis are done using bioinformatic tools to provide the sequence of the target DNA to database library available. The identification of the bacteria can be achieved and we can now know the exact genus and taxonomy of the bacteria based on the comparisons.
Next, we move to the next part, which is the benefit of 16S rRNA sequencing. So for the first benefit is it has high bacterial coverage. 16S rRNA sequencing is used to identify the species of bacteria based on its 16S gene, and all bacteria contain 16S gene. That's the first benefit. And then the second benefit is it's require a low amount of DNA input. 16S rRNA sequencing is very sensitive, which is only require 1 femtogram, which is only 10 copies of 16S rRNA gene of bacteria. And then we move to the third benefit of 16S rRNA sequencing, which is it has a low risk of false positive. 16S rRNA sequencing being able to detect the closely related genome. This closely related genome might be from the different species but in the same genus, or both of them from the different genus. And then we move to the fourth benefit, which is it is low cost compared to other sequencing method. 16S rRNA is the cheapest.
What is shotgun metagenomic sequencing? Shotgun metagenomic sequence refers to a technique that is performed for taxonomic profiling, include diversity, abundance, as well as functional analysis. In shotgun sequencing, DNA is broken up randomly into numerous small segments, which are sequenced using the chain termination method to obtain reads. Multiple overlapping reads for the target DNA are obtained by performing several rounds of defragmentation and sequencing. For your information, data obtained can be used for the additional analysis. The first analysis is metagenomic assembly and binning. What is metagenomic assembly? It is the stitching together of individual DNA sequences into genes or organisms, while binning is the process of grouping metagenomic sequences by their organism of origin. In metagenomic studies, binning allows the reconstruction of known and unknown genomes, so it will enable a broad description of the community and creating a starting point for further analysis of the organism. The second analysis is metabolic function profiling. The metabolic profiling is the measurement in biological systems of complement of low molecular weight metabolites and their intermediates. Metabolic profiling is most often performed using either nuclear magnetic resonance, NMR, or mass spectrometry, MS. And the final test is the antibiotic resistant gene profiling. So what is antibiotic resistant profiling? Antibiotic resistant test, also known as antimicrobial susceptibility testing, is the laboratory testing performed on the bacteria to find out if they are resistant to one or more antibiotics.
Shotgun sequencing begins with DNA extraction. Then the DNA will be fragmented into a smaller component that are independently sequenced. This independently sequenced fragment will be aligned to loci for the multitude of a genome in the sample. There are two analysis methods that could be done, such as marker gene analysis and binning analysis. Marker gene analysis takes the sequence and compare them to a database of known taxonomically informative sequence. Meanwhile, in the binning analysis, there are multiple methods such as compositional binning, fragment recruitment, and similarity binning. Sequences are then compared to phylogenetic and functional database to obtain the taxonomy and functional profile.
By utilizing the shotgun metagenomic sequencing, researchers are able to comprehensively sample all genes in all organisms present in a complex sample. This enables them to evaluate the bacterial diversity and detect the abundance of microbes in various environments. Therefore, the researchers can simultaneously identify and profile species from all taxonomic domains, including viruses, which is useful for microbiome research. This is because the technique can read all genomic DNA in a sample, unlike the 16S rRNA implicant sequencing that just can read one specific region of DNA in bacteria. In addition, shotgun metagenomic sequencing can identify microorganisms at the species and sometimes at the strain level resolution. Therefore, for a deeper look at the species or strain of the microbe, these techniques are more suitable for the research. As genome sequence, microbial genes observed in the sample can be identified and profiled, providing additional information about the microbiome functional potential. For example, genes associated with the hydrocarbon degradation pathway and the DNA repair pathway were found within the microbial community in the hot spring sample and appears to be important for the survival. Therefore, by profiling the microbial genes, the researchers are able to identify the microbe community and also the ways on how they function or make their living in the extreme environment.